Hello everyone, welcome and thank you for dialing in for our webinar today. My name is Xuan Chunling. I'm an R&D manager at GenScript ProBio and responsible for viral vector manufacturing process development. Today, I'm going to talk about process development strategy of Adeno-associated Viral Vector, which is also known as AV. Okay, let's begin. Today's webinar includes the following four parts. Firstly, I'll introduce the market perspective of AAV in the gene therapy, and then I'll give you a brief introduction about the basic structure of AAV and how it works. Followed by manufacturing optimization strategy, I will also share some successful data. Finally, what we can help to accelerate your gene therapy project will be included. As you all know, gene therapy is a technique that modifies a person's gene to treat or prevent disease. There are various types of gene therapy products, which can be divided by the delivery vehicles, such as AV, ADV, and non-viral vector. According to clinical trial data, AV is the mostly investigated delivery vector, as it is not known to be associated with any human pathology. For gene therapy, AAV containing the gene of interest is directly injected into the patient, and the multiple copies of the protein are produced by the patient's cell. Here, I would like to talk about the milestones of the approved AAV drugs. AAV was first discovered as a contaminant of adenovirus preparation in 1965. After a few decades, AAV is used in the clinical trials. The first AAV gene therapy drug, Glabella, was approved by the EMA to treat lipoprotein lipase deficiency in 2012. And the first AAV gene therapy product, Luxturna, licensed in the United States, followed 5 years later. And now, a large number of gene therapy products delivered by AAV have been developed and bring hope to the next generation therapeutics. From the market perspective, you see the AAV market is expected to reach $471 million by 2025. Besides, the market share of AAV will also increase in the whole global vector market. Therefore, you can actually see the demand for AAV manufacturing is growing. Until now, numerous AAV cell types have been identified. Different cell types of AAV target different organs. There are 3 vectorized AAV cell types that have gained regulatory approval. That have gained regulatory approval for commercial use in patients: AV1, AV2, and AV9. AV8 and AV5 are increasingly being utilized as novel and more efficient capsid. A large percentage of AAV gene therapy under clinical development is focused on the CNS, including brain and the eye. The second part is a basic introduction of the AV. Basically, AV is a single-stranded DNA virus. One type AV requires another virus, such as an adenovirus or herpes virus, to replicate. The AV genome is 4.7 kilobase paired. Kilobase pairs flanked at both ends by structural elements known as the ITR. The ITRs are required as a packaging signal of genome encapsulation after replication. The AV genome is highly compact with complex overlapping coding regions. The AV genome harbors two main genes, RIP and CAP, which encode 9 distinct protein products. All 4 RIP proteins have common helices and ATPase domains that function in replication and encapsulation. When cells are infected with the AV, the first step in reproductive replication is conversion of the released ssDNA into double-stranded DNA template for transcription. Here you can see the transduction pathway of AV. Firstly, AV is recognized by the surface receptor of the host cell, and it triggers internalization. Owing to the somewhat low pH environment of the endosome, AV, the VP1 and the VP2 regions of AV undergoes a conformational change. Following endosome escape, AV undergoes transport into the nucleus and encoding. Replication and the packaging of AV is dependent on host cell factors and elements provided by the hypervirus, such as HSV and ADV. The middle figure illustrates the host and the hypervirus proteins that interact with AV genome. Okay, for this part, I'd like to show how the AV integrates. Wild-type AV integrates into the human genome at a specific region on the chromosome 19. However, the recombinant AV cannot be integrated into the host genome, and the abnormal genes can only be replaced by the homologous recombination. After talking about the basic information of the AV, in the next part, I'll talk about the optimization in the AV manufacturing. In general, there are two modes of vector production: transient transfection system and stable cell lines, which may be mammalian or insect cells in case of baculovirus vectors. Manufacturing viral vector by transient transfection system offers advantages and disadvantages. It is flexible and efficient, does not require development of stable cell lines. However, transient transfection has disadvantages due to the requirement of costly plasmid for each transfection lot and the possible requirement of additional purification step to remove plasmid. Moreover, transient transfection of suspension cells at large scale can be difficult and can result in relatively low yield. The flowchart I showed here is the general process for AV production using triple transfection. Generally, we divide the whole manufacturing process into upstream process and downstream process. Approaches to vector purification exploit the physical characteristic of viral particles, such as size, surface charge, and hydrophobicity. Viral vectors vary greatly in physical characteristics, depending on the type of virus. Since AV is partly released from the cells, AV is generally harvested by cell lysis to improve yield. Cell lysis can generate significant quantities of host cell contaminants, including DNA and protein, that increase purification burden. Okay. This figure illustrates the requirements and improvement opportunity with an upstream process. Cells used in an approval process for human medicines need to be fully documented and traceable to their origin, and need to adopt it to long-term stable expression of the target product with its desired features. Attention needs to be paid to the quality attributes of the product and the capability of the cells to synthesize it without changes to these attributes. For the downstream process, the most important is the recovery steps need to isolate the product and prepare the process feed stream for purification by removing cells, debris, and cell culture components. In addition to mitigating the risk associated with the impurities, the downstream process needs to mitigate the risk of contamination. A good downstream process should remove all impurities that can potentially alter or damage our product as early as possible. Okay, to comprehensively consider the whole process and to achieve process capability supporting consistent product quality, good yield, and economic efficiency, choices and actions in the upstream have obvious impact on the downstream product recovery and purification steps. As a polish to integration efforts, the process will be simplified and wasteful activity will be reduced to an absolute minimum. Here I showed the quality attributes of our AV. As you may know, AV product will be directly injected into the human body, so the AV product must meet rigorous safety guidelines. This is an overview of analytical methods used in viral vector manufacturing, including virus particle quantity, identification, potency, purity, and safety. Because AV has empty capsid, VG titer and capsid titer will be measured respectively. In the aspect of purity detection, in addition to detecting the impurities related to the process, AV empty ratio will also be detected. Now, I want to share some actual cases about what we have done to optimize our AV manufacturing process. Here we optimize the transfection parameters during upstream process. The 3 packaging plasmids were mixed with the transfection reagent. Since the transfection reagent was positively charged, the plasmid can break through the electrostatic of the cell membrane and enter the cells. AV packaging elements were synthesized in cells and assembled into complete virus particles. Thus, during this process, plasmid ratio and concentrations, transfection reagent, and cell densities are the key parameters of AV transfection. As I mentioned before, the serotype 2, 5, 8, and 9 are most widely investigated, so we mainly focus on the process development of these 4 serotypes and optimize the key parameters by DOE to improve AAV titer in different serotypes. You can see our crew titer is significantly increased after optimization. Then we explore our method in scaling up the manufacturing process. We scaled up from 30 milliliter to 25 liter bioreactor. The crew titer shows good consistency from small scale to large scale, both in shake flask, glass bioreactor, and wave bioreactor. An average titer of the crew acid is around 2 times 10 to the power of 14 Vg per liter. And for the downstream process, the optimization of downstream process parameters, including nuclease treatment, TFF concentration, affinity chromatography resin selection, and IEX elution conditions. For TFF, we have compared 3 kinds of hollow fiber cutoff molecular weight by comparing the residue impurities and the recovery. It is finally to determine to use the 100 kD hollow fiber. As you know, host cell DNA is a common process-related impurities encountered when manufacturing viral vector for gene therapy. Due to the safety issues, the maximum amount of DNA allowed in the final dose administered to humans are set to 10 nanogram per dose. We have determined the optimal reaction conditions for nuclease treatment. We also compared different affinity chromatography resin and chose one of them based on the recovery and the impurity removal rate. AV vector can produce a significant portion of empty capsid, devoid of transgene at up to 95% to total particles. IEX is the one of the few methods that can separate full capsid from empty. We can separate the intact AAV from the empty particles by optimizing the elution conditions. One of the major considerations of moving to clinical manufacturing is how to employ increased production scale. At larger scales, the purification method needs to accommodate large volumes and with high efficiency and speed. We have completed 3 batches of 2 liter and 1 batch of 25 liter. Average of overall recovery is 24.84%, and the batch yield is around 10 to the power of 13 Vg per liter. Okay, to sum up, after all the parameters have been optimized, actually we got quite good result to produce AV5. For the upstream process development, it could be scalable from 30 milliliter to 25 liter. The crew titer is up to 2.5 times 10 to the power of 14 Vg per liter, which is above industry grade. And for the downstream process, the recovery rate between 30% to 60%. In line with industrial grade. Okay, these are the critical quality analysis data. During manufacturing of AAV vectors, a high percentage of capsid might not incorporate of any transgene or contain fragments of the transgene. The presence of these impurities could increase immunogenicity or inhibit transduction of full capsid by competing for vector binding site on cells. We use TEM and AUC to detect AV empty capsid ratio. As you know, AUC is an efficient and reproducible method that have been standard in quantifying AV empty ratio to quantifying AV empty to full capsid ratio. Although the clinical success of AV gene therapy is encouraging, we must acknowledge the limitation and the challenges of this gene delivery platform, which includes issues with AV manufacturing and the immunological barrier to delivery. There are several areas of improvement for AV and for gene therapy. The typical AV genome design carries a gene expression set, which includes a promoter, a transgene, and a transcription termination signal. Together, these components serve the main function of expression therapeutic level of gene product once the vector is delivered to the target cell nucleus. A novel capsid are engineered to gain new properties and characteristics. Capsid development approaches can be divided into three main categories: natural discovery, rational design, and directed evolution. And to overcome the limited scalability due to the use of anchorage-dependent cell cultures, a suspension-based transfection process, as well as 3 different scalable AV vector production systems have been developed. Last but not least, I'll briefly introduce what we can help now. GenScript is a global biotech company and is divided into 4 segments. And we are the GenScript ProBio, focused on biologics CDMO. GenScript ProBio proactively provides end-to-end service from drug discovery to commercialization with professional solutions and efficient processes to accelerate drug development for customers. Through all these years of development, we have completed 60 plasmid and virus CMC and clinical projects, 7 IND approved from NMPA and FDA, and we have conducted over 20 CMC projects. We also provide plasmid and viral vector service for over 30 big pharma and leading biotech. Currently, we provide from plasmid manufacturing to AV CMC study and manufacturing services, and we can support your projects in the whole drug development circle. We have prograde for preclinical study, CMC study for IND filling, clinical GMP grade for early phases, and cGMP grade for late clinical phase and commercialization. Okay, thank you for your attention, and that's all what I've got for you here today. And thank you.